Welcome back to this next video in which we are discussing HPVs and cervical cancer. Okay, so we've now had a basic introduction to human papillomaviruses. What we now want to discuss is the life cycle of human papillomaviruses. We want to discuss how are these things going to infect squamous stratified epithelia, and how are they going to use the squamous stratified epithelium to create more virus particles. So remember, the aim of this virus life cycle is to go from having one virus particle to having loads of virus particles. And it's important when studying a life cycle of a virus or indeed of another pathogen to remember that that's always the aim, to create more. Okay, so we are going to discuss this life cycle, and this life cycle is going to be applicable. What we're going to say is going to be applicable for the life cycle of all the different HPVs, whether we're talking about a cutaneous or a mucosal HPV. Even though they infect different types of squamous stratified epithelia, the physiology of the cutaneous versus the mucosal squamous stratified epithelia is similar enough that the life cycles are very much so comparable. So at our level of detail, everything that we say is going to be applicable for all the different human papillomaviruses. Okay, so I'll just put a title here. We are going to discuss the human papillomavirus life cycle then now. So we are going to begin this story from just having a virus particle at the squamous stratified epithelium that it's going to infect. And of course, if it's a cutaneous HPV, it'll be cutaneous squamous stratified epithelium. If it's a mucosal HPV, then it'll be a mucosal squamous stratified epithelium. We are not at this stage going to discuss transmission. I think it's best to describe the entire life cycle and then at the end talk about transmission rather than at the beginning talk about transmission. So don't worry, we will have the delight of talking about HPV transmission, but we'll do it right at the end. So let's begin then just by drawing the virus particle. So we'll draw our little virus here. So this is going to be the capsid here in blue. So this is going to be our human papillomavirus here. And of course inside the capsid you'll then have the episome, and I'll have the episome, oh sorry, not the episome, that's a piece of terminology I have not introduced you to yet. The genome, I should say. The genome, which is circular double-stranded DNA, is there at the centre of the capsid. So here is our human papillomavirus, a capsid with the double-stranded circular genome at the centre. So it's now going to infect its squamous stratified epithelium, and as I say, if it's a cutaneous, it will be a cutaneous epithelium. If it's a mucosal HPV, it will be a mucosal epithelium. So the first question which arises is, which cell in the stratified squamous epithelium does the human papillomavirus infect? Now the obvious answer would be to say, okay, well, it will be the most superficial one, because how else is it going to get to any of the other cells? The answer is no, it does not infect the superficial one. It would be no use to infect the superficial one because the superficial ones aren't particularly functional. I mean, if we're talking about a superficial cell in a keratinized stratified squamous epithelium, that doesn't even have a nucleus anymore. It's completely overloaded in keratin. The virus isn't going to have much use for that type of cell. So it does not infect the most superficial layer of the squamous stratified epithelium. Instead, it has to infect the basal cell layer of the squamous stratified epithelium. So let's start by drawing this in our picture here. So we'll start, of course, by drawing the basement membrane. So in red here, this is going to be our basement membrane, and let's now draw on one of the basal cells here in white. So this is going to represent a basal cell drawn nice and big now. And here, this is going to be the nucleus of our basal cell. Okay, right, and I'll just label it up as a basal cell here. So of course, this is all well and good, but the question which arises is, well, how on earth does it get access to the basal cell? And this is where I finally answer the question of why is the cervix so vulnerable to HPV infection? So, In order to get access to the basal cell layer of a squamous stratified epithelium, the normal thing that we think has to happen is you have to get a little injury to the squamous stratified epithelium. So we think that the normal way that this happens is through what we call a microabrasion. So a little injury in the skin that makes a piercing hole, or not just the skin, in one of these squamous stratified epithelia, that makes a little hole that goes all the way down to the basal cell layer, and then of course the virus can go through that microabrasion and get to the basal cell layer. So that's one of the major ways that we think HPV infections can infect the basal cell layer. And now let's talk about the cervix. So with regards to the cervix, we have to remember that metaplasia is occurring. So cervix metaplasia is going to be the other way that HPV infections can get access to the basal cell layers. So you remember when I was describing metaplasia to you, remember what happens is that the columnar epithelium turns into a squamous stratified epithelium, and this happens through the emergence of those subcolumnar cells that are going to become basal cells, and they're going to give rise to all of the layers above the basal cell layer in the squamous stratified epithelium. However, in the metaplastic process, there is a time when the subcolumnar cell is exposed to the vaginal environment and has not yet produced all of the layers above it, i.e. in incomplete metaplasia. So I should put that as an important term. So in incomplete metaplasia, where you've just got the basal cell that has formed from a subcolumnar cell and it has not yet produced the layers of cells on top of it, then it will be exposed to the vaginal environment, and then it is at high risk of getting an HPV virion particle infect it rather. So this is why the cervix is so vulnerable to HPV infection, because as the metaplastic process is occurring, and we've gone through the fact that it occurs over decades and decades and decades, so you are vulnerable for decades and decades and decades. As that process is occurring, there are times where the subcolumnar cells that have become the basal cells that will make squamous stratified epithelium have not yet made the layers on top of them, the midzone and the superficial zone yet, and therefore are exposed, and then the HPV virion particles can infect them. So this incomplete metaplasia, this is the reason that the cervix is so vulnerable to HPV infection, because we have the subcolumnar cells which are becoming basal cells which have not yet produced the protective layers on top. Elsewhere, for the other squamous stratified epithelia, the way that we believe HPV virion particles get access to the basal cell layer is through microabrasion. Okay, so there's the starting point of the story, how the HPV actually gets access to the cell that it's firstly going to infect. And I'll say something that I've already said in previous videos, which is the fact that human papillomaviruses do not cause systemic infections. They only affect these squamous stratified epithelia. They do not go underneath the basement membrane and cause infection of the tissue underneath. They just infect the squamous stratified epithelium, so they do not cause systemic infections. Right. So, the next thing you're probably waiting for me to tell you a fantastic story of is how does the human papillomavirus get into the cell? So, you know, if we're talking about HIV virus or influenza viruses, we have a fantastic understanding of how those viruses get into the cells. In the case of human papillomaviruses, I'm afraid we do not have that same sort of understanding. We do not have a good understanding at all of how human papillomaviruses get inside the cell. What we do know is that it takes hours and hours and hours for the human papillomavirus, having got to the basal cell, to actually get into the basal cell. And that little fact that I've just told you there is actually going to be crucial, we think, to why human papillomavirus vaccination is actually effective, but you'll have to wait right until the end of the video to hear more about that. So, coming back to the entry, we know it takes hours and hours and hours, but we do not know what receptors on the cell are important really at all. It's still very, very controversial, and annoyingly it seems as though it might be different for the different types of human papillomavirus. However, it's still not very well understood at all, so I am going to draw an arrow to make up for the lack of a story here. So I'll do it in this green colour here. So what's going to happen is the virus is going to enter the cell and it's going to unpackage, and overall the end point of the story, the end point of the arrow, is that the genome of the human papillomavirus ends up inside the nucleus of the cell, and now that it's inside the nucleus of the cell, it is called an episome. So that piece of terminology that I accidentally used earlier on when describing the genome inside the human papillomavirus, I'm now using it here. So once the genome is inside the nucleus of the basal cell of the squamous stratified epithelium, it's described as an episome. So this, I'm afraid, is the arrow that I'm drawing for entry. I can't tell you any more than that. That is as much as I know. So I've reduced it all down to an arrow. What happens is the virus gets into the basal cell, and overall the genome ends up inside the nucleus. So it gets to the basal cell, and now I've drawn a great big arrow across this. Nucleus gets into genome is what I know. That's all I can say about this. Okay, so we've now then got the viral genome inside the nucleus of the cell as an episome. What we now want to talk about is what's going to happen next. So, the human papillomavirus genome, we know that it has 8,000 bases. We also know that it codes for 8 genes, E1, E2, E4, E5, E6 and E7, the 6 early proteins, and L1 and L2, the 2 late proteins. Now, none of those proteins that the viral genome can actually make are RNA polymerases or DNA polymerases. It does not code its own nucleic acid polymerases. So, for transcription, and indeed for protein synthesis altogether, it is completely reliant on the host cell machinery. In addition, for DNA replication, it is completely reliant on the host cell machinery. Contrast that to many viruses which code their own DNA polymerase so that they can divide their own genome without being reliant on the host cell division. You see, that's the problem for human papillomavirus. Its genome needs host cell machinery in order to replicate. Now, host cell machinery for DNA replication is not normally active. It's only active when a cell divides, and that means that the human papillomavirus needs the cell that it has infected to be dividing in order for it to be able to copy its own genome. And that hopefully starts to give you the picture of why these things might lead to the formation of warts and why they might lead to the formation of cancer, because they're going to stimulate cells to divide. However, we'll spell this story out in much more detail as we go on. So, what's the first thing that happens? The first thing that happens is that the E1 and E2 genes are going to actually be expressed. Host cell protein synthesis machinery is going to be used by the virus to make the E1 and E2 proteins. And E1 and E2 are utterly essential for the viral genome to be able to be copied. So let me put these proteins in. So what's going to happen then is here in the little yellow blob here, this is E1 protein, so you're going to get E1 being produced. You're also going to get E2 being produced, and I'll just have E2 in blue here. So this is the E2 protein. So this is the first stage in this process. The viral episome, now that it's inside the nucleus of the basal cell, is going to use host cell protein synthesis machinery to make the E1 and E2 proteins. So let's now talk about what E1 and E2 do. E1 and E2, if you don't have those, the viral genome cannot be copied whatsoever, because they are the things which bind to the origin of replication and which recruit the host cell DNA polymerase to the origin of replication for DNA replication to occur. So let me just draw a bigger picture of our genome here. So let's imagine that this is the viral genome, so this is the double-stranded piece of circular DNA. Now, a certain portion of this genome, let's say it's this portion up here, is going to be the origin of replication. So just like in human chromosomes, you have an origin of replication in this viral genome, which is where the DNA polymerase enzyme is going to bind onto in order for it to then begin replication. So this is exactly like in human chromosomes, so origin of replication here. And for short, we will just abbreviate origin of replication down to ORI. Okay, so the host cell DNA polymerase, the human host cell DNA polymerase, cannot just bind to the viral origin of replication. First, it needs E2 and E1 to bind onto there, and then the DNA polymerase can bind on. So the first one to bind onto the viral origin of replication is the E2 protein, and it recruits E1. So there we go, and this then is called the E1, E2 complex. Okay, so for the viral genome to be copied, you need E1 and E2 complexes to bind on to the origin of replication, and then they can recruit the DNA polymerase machinery of the host cell onto that origin of replication, and then replication can begin. So these proteins are going to go back into the nucleus, they're going to bind onto the origin of replication, and now we're ready to actually replicate the genome of our virus. So it still requires, however, the host cell DNA polymerase machinery to be active. So therefore it requires the basal cell to divide. And I just want to repeat this message because it is so important. The virus does not code its own DNA polymerase. Lots of viruses do code their own DNA polymerase to get round this problem that the human papillomavirus faces. Because human papillomavirus does not code for its own DNA polymerase, it relies on using the host cell DNA polymerase. Now, the host cell DNA polymerase is only going to be active when the host cell is actually dividing itself. So the viral genome can only be copied when the cell is dividing. Now, this is fantastic because the basal cell, of course, it is dividing. It does divide. We know this. We've gone over this physiology, so it does divide. So the viral genome is going to have the opportunity to be copied. So what's now going to happen then is the basal cell is going to divide, and it will produce another stem cell that will remain attached to the basement membrane, and then a daughter cell which will move up into the mid-zone layers. So I will draw this daughter cell in purple here. So here is the other cell that's produced, the other daughter cell, and I'll give it a nucleus here. Okay, so what's going to happen is we're going to get cell division, and we'll produce the daughter cell and another cell that will remain in the basal cell there. So this layer up here, this is representing the mid-zone of the squamous stratified epithelium. And I know, of course, the mid-zone would have far more layers of cells than just one, but for a basic picture, here we go. Okay, now, when the basal cell divides into two cells, one which will move up into the mid-zone and one which remains in the basal cell layer, the viral genome can also be copied. The DNA polymerase, once it's activated, can bind to the E1, E2 complex and replicate the viral genome, and you'll have two copies of this episome now. Now, when the cell divides, when the host cell divides, of course all of the chromosomes in the host cell will be copied as well, and they'll only be copied once, so that each of the daughter cells gets one copy. However, the viral genome has no such limit. It can be copied far more times than just once, so it is not the case that when the cell divides you only copy the viral genome once so that both cells get a copy. Instead, you're going to copy it multiple different times, and this is why it's important to produce more E1 and E2 than just one copy each, so that once you've got more copies of the genome, the E1 and E2s can form on their combined, the E1 and E2 complexes can form on their origins of replication as well, so that you can get them dividing as well. So, in this single division then, you're going to produce lots of episomes. So I'll just draw this on here. So here's another episome, here's another episome, here's another episome. So you produce lots of episomes. The episomes are copied far more rapidly than the host cell DNA is copied. The host cell DNA is only copied once, so that each daughter cell has one copy of each chromosome. The viral episome will be copied more than once, and this means that both of the daughter cells will end up with lots of copies of the viral episome, just like what I've shown here. So both daughter cells now end up with lots of copies of the viral episome. Okay, now this is still what is called the low division rate phase of the viral life cycle. So this portion that I've just gone over, this is the low division rate phase of the life cycle, because of course we are amplifying the viral genome here. We are making more copies of the viral genome. However, this is not the major production part of the viral life cycle. Later on in the mid-zone, as I will come to, this viral genome production gets out of control and you produce loads and loads of them. So this is just the low division rate phase of the viral life cycle. We are producing a few more viral genomes, but this is not the major productive part of the life cycle yet. It gets more dramatic than this. So we still have only a few viral episomes in each nucleus of the two daughter cells. We don't have thousands yet, whereas in the high production part you'll end up with thousands of viral genomes being produced inside each cell. Okay, so the next portion of the life cycle, and I think actually let's just summarise where we've got to now and then we'll move on to the next part. So we started off with human papillomavirus coming in and getting to the basal cell, and remember we talked about ways that it can actually get access to the basal cell, either through microabrasion of the squamous stratified epithelium or in the cervix, where we've got incomplete metaplasia, the basal cells can actually be exposed to the environment and therefore human papillomaviruses can quite easily get into them. We discussed the fact that the entry of the virus is not well understood at all. We know it takes a very, very long time, and we'll come back to the significance of that later on. But we don't know very much about how it gets in at all. So what we did is we drew an arrow from the virus to the genome getting into the nucleus. That's the next stage that we moved on to. So the genome is now in the nucleus. It's called an episome. And the first thing it's going to do is use host cell machinery to make loads of copies of the E1 and E2 protein. And the importance of the E1 and E2 proteins is that without them, you cannot copy the viral episome whatsoever. They are needed to recruit the host cell DNA polymerase to the origin of replication. I think I will just put in here that now that they are present, you can recruit the host cell DNA polymerase. So DNAP stands for DNA polymerase, DNAP. Okay, and that will allow the replication of the genome. However, there's still a problem, which is that you need the activation of the host cell DNA polymerase, and that's only going to occur when the cell is dividing. Now, luckily, we've infected a basal cell, which will be dividing. Therefore, when the basal cell divides into two daughter cells, one that remains a basal cell and one that becomes a mid-zone cell, you will be able to copy the viral episome whilst the DNA polymerases are active and copying the host cell genomes. And we discussed the fact that the episomes will be copied at a faster rate than the host cell chromosomes. The host cell chromosomes will only be copied once, whereas in the same period, the viral episome will be copied more than that number of times. So you'll get quite a few viral episomes in the nucleus of both daughter cells here, but it's still a low number. We call it a low copy number. So I might actually put that as a key term as well. So we say that the virus is present in these cells at a low copy number, and this is the low division rate portion of the life cycle. Later on we will see the high division rate portion of the life cycle, where you produce loads of viral episomes in the nucleus of a single cell, and that's the high copy number portion of the life cycle. Okay, so what happens now? Well, of course, this basal cell will divide again, produce more daughter cells, which again will have more episomes in. So that's fantastic. We've got the viral episome in these basal cells, and that means that every daughter cell that this basal cell now produces that's going to go up to the midzone will also have viral episomes in. So this is looking jolly good. Now what we need to turn to is what's going to happen in these daughter cells that are in the midzone then. Okay, well, remember, normally what now happens is these cells differentiate, and indeed I've already shown this cell differentiating. It has changed from the basal cell here. So it begins this differentiation process and it gradually changes as it goes up. And remember, I told you that when it undertakes this differentiation process, it does not divide again. Now that's useless for the human papillomavirus, because that would mean that the maximum number of virus particles it could produce are the ones corresponding to the genomes that it's already got from the division of the basal cell that produced this cell. So you wouldn't be able to produce any more because you have to have this cell dividing, you have to have the activation of the DNA polymerase machinery for the viral genome to be able to be copied. So what's going to happen is the human papillomaviruses episomes are going to hugely disturb this differentiation process. They're going to put their foot down on the accelerator and they're going to say, no, you are not going to stop dividing, you are going to divide anyway. We are going to activate your DNA polymerase machinery ourselves. So what happens now, once the cell has differentiated somewhat, and we don't fully understand how this works, but once the cell has begun the differentiation process, you start expressing the other early proteins to express E4, E5, E6, and E7. You also produce even more E1 and E2, and we know that E1 and E2 have already been expressed, but you're going to produce even more of them. So you're going to produce all of the early proteins now. Now, the reason we're producing more E1 and E2 is because, of course, to copy the viral genomes, which is what we're aiming to do, we're aiming to make loads more viral genomes, we're aiming to go into the high division rate portion of the life cycle. In order for that to happen, we have to have more E1 and E2. Without E1 and E2, you can't copy the viral genome. So that makes sense that we're producing more E1 and E2. What are these other early proteins going to do? What are E4, E5, E6, and E7 going to do? Well, we don't really understand very well what E4 and E5 do, so I'm not going to discuss those any further. However, E6 and E7, these are two very, very famous proteins. The E6 and E7 proteins of human papillomaviruses are famous for all the wrong reasons. They are famous in cancer textbooks because what these proteins together do is they make these cells that should not divide again. In fact, I will actually just put that as a little annotation. This cell should not divide again. It should be differentiating, not dividing. And indeed, the two processes don't go together. If you're differentiating, you are not dividing. If you're dividing, you are not differentiating. So this should not divide again. But these proteins are so powerful, E6 and E7 together are so powerful that they're going to force this cell, they're going to hijack all of the machinery inside the cell and force it to divide at a very, very fast rate so that the viral genome can use the host cell DNA polymerase to copy its genome. So we will see that E7 inhibits the tumour suppressor protein retinoblastoma, and E6 inhibits the P53 protein, two of the most famous tumour suppressor proteins known. I mean, P53 is probably the most famous tumour suppressor protein known. And together, these two actions are going to lead to this cell completely over-dividing. Okay, so I think we'll have a break here. In the next video, what we will talk about is E7 protein. We'll start off with E7 protein. Now, I know this isn't in numeric order, but I think the logical way is to start with E7 protein and then come back and discuss E6 protein, because E6 protein's action is to stop response that would happen if you just had E7 proteins action alone. So we'll do E7 and then E6, and we'll understand how the presence of the viral genome inside these mid-zone cells is going to lead to them dividing at an incredibly fast rate, and therefore is going to lead to them producing loads of copies of the viral genome, i.e. the high division rate phase of the viral life cycle. So we'll do that in the next video.